thank you, and good morning, everybody, and thank you to all that put the effort into getting this put together and inviting me to share a little bit about the work that we have going on here in the department. So just to tell you up front, the talk that I'll have here today is slightly different from the previous two, and probably the rest of them that you'll hear today, where you hear a lot of things that you can do right now to make improvements on your farm. What we'll have here is stuff that you won't be able to take it home today, perhaps, but hopefully in the near future, the work that we're doing now will be something that you'll be able to use on your farm to be able to make improvements to solve some of the issues that are taking place on your farm. There's two projects that I'm going to talk about today that were funded by the milk checkoff funds, and both of them deal very similarly to the use of vitamin D. But first, I really want to emphasize how critical these funds are to be able, for us as researchers, to be able to work on solutions for the dairy industry. A big thanks to the Milk Checkoff Board for making these available. So there's two key projects that I have. One is I was the lead investigator on, and this is entitled Minimizing Cost of Mastitis Through Enhancing Antimicrobial Production in the udder. The other is one that Dr. Santos was the lead investigator on, and this is using a form of vitamin D, the active form of vitamin D, to maintain blood calcium in postpartum cows and subsequently enhance immunity. So really overall our goal for Dr. Santos and I and many researchers here in the department in the vet school is to improve transition cow health. That's really been identified as a major inhibitor to a good production of cattle. And so this is our goal is to minimize the incidence of disease in transition cows. And obviously there's been work over the years that have demonstrated that proper nutrition, proper management can really make huge advancements in minimizing the diseases in those transition cows. But you may be out there doing everything right, doing everything that we've recommended for you to do in terms of nutrition, management, etc. You're still going to run into problems. Even the best nutrition and management isn't going to solve all the problems. And so that's where we recognize those issues, and we're looking for ways to make advances, find the solutions to that. So that's what a couple of these research projects are addressing. So kind of a general theme to our approach is identify viable therapies that really we hope would be useful that we can take to the dairy farm someday and improve transition cow health. So this encompasses kind of a series of work that goes on. Initially we have the basic research that's going to take place that really we use to identify how does the immune system of the cow work. What are the signals regulating the immune system? What can we use to manipulate that immune system? And then that works along with the applied research that we do. How can we take those basic fundamental findings that we make in the laboratory, take them to develop useful therapies that you can, approaches that you can use on the farm. And vice versa, this goes back and forth. Much of the applied work that we do helps lead to more basic research, more basic questions that we need to go back and answer. And it's a continuous cycle that we use to help continue to improve production of the dairy cow. So the two projects that I'm going to talk about here today, the big focus is on the improving immunity, enhancing the immune system to really the ultimate goal is decrease disease incidence or improve resistance to disease. So targeting the immune system. And it focuses on these two molecules here, calcium and vitamin D. Now it's not just as simple as making sure the cow has enough vitamin D and have enough calcium. There's a very strict control of both of these that we really need to understand the physiology of the cow and how they're regulated in the cow. So this adds in a little bit more of the complexity that's involved in how vitamin D and calcium are used. And what we've discovered or scientists have discovered quite some time ago, actually about 50 years ago now, that there's this hormone here, this molecule here that's derived from vitamin D. It's called the 1,25-dihydroxyvitamin D. This is the active molecule of vitamin D, and it has to be activated in the within the cow, and the activation of that molecule is very tightly controlled. Well, we've made some discoveries recently that this molecule of vitamin D can act directly on the immune system, and we also know that it acts on calcium, and we've also made discoveries that calcium is critical for proper immune function. And one of the problems that develops in transition cows, obviously, is the hypocalcemia that develops. So are there, the questions that we're asking then, are there ways that we can use this active form of vitamin D to either through calcium or directly through the immune system to enhance immunity of the dairy cow? So this is some data from discoveries that we made in the laboratory several years ago, showing how vitamin D is used in the immune system. And don't get confused with everything that's going on here. The main point is this is a major form of vitamin D that's in the blood, and the immune cells have the ability to convert that to the active form. And what we found is that when they produce this active form, they're able to enhance one of these antimicrobial or bactericidal molecules called nitric oxide. So if we add more of this molecule here, metabolite of vitamin D here, to the cells in culture, we get an increase in this nitric oxide production. Another one that one of my graduate students, Catherine Merriman, discovered while she was here just a couple years ago, was this production of another bactericidal molecule. It's called the beta-defensin. It's a small protein that's produced by the cells, the immune cells in the cow, and we found that treating cells with this 25D molecule here in the laboratory, we could increase the production of this beta-defensin protein. So, we're able to show this in the laboratory, we're able to do this in a dish, but that really doesn't mean anything to everybody out there, because you want to see, does this actually happen in the dairy cow? So this is kind of the basis leading up to one of our milk checkoff projects, and that is, can we actually increase through vitamin D, can we actually increase the production of some of these bactericidal molecules, antimicrobial molecules, in the dairy cow? So we thought, well, what if we use this active form of vitamin D and go directly into the udder and see if we can get an increase in some of these responses within the udder itself. So we designed an experiment where we used intramammary treatments of this active vitamin D molecule, the 1,25-dihydroxyvitamin D, or a placebo. So we did this, we used the quarter as the experimental unit. So this is the two treatments were within the quarter, within each cow itself. So we did these treatments, and then we'd, within either 4 or 8 hours later, we'd isolate the milk somatic cells. And from there, we can use those milk somatic cells to measure immune responses. And we do this by measuring gene expression. And that's just one of the parameters that we're able to use to identify, whether or not there's a change in immune status or immune competency of the cells there. So here's kind of the key results of one of the first project that I'm going to talk about here, and that's we took cows, treated them with the intramammary with this vitamin D molecule, the active vitamin D molecule, or a control treatment. So here in the blue is the active vitamin D treated quarters, and then the orange or red is the control treated quarters. And so and then these are expression of several genes that related to bactericidal capabilities of the cell. So really it signifies whether or not we have an increase in any of these parameters related to antimicrobial protein production. And so we can see from doing this, we have a couple of them. This is a nitric oxide synthase. It produces nitric oxide, a measure of the immune response. And we see an increase there fairly rapidly within 4 hours after treatment. There's several of these beta defensin molecules that I told you about as well. We didn't see a change in several of them, but there was one of them, this beta defensin 7, that we did see an increase in with the intramammary 125B treatment. So this particular experiment was all done in healthy cows, no mastitis or anything. So the next question is, do we actually see an increase in cows that would have mastitis? Oh, this is I forgot I had this in here. This is just focusing in on the changes within nitric oxide production and the beta defensin 7. So showing an increase in both of these markers fairly quickly after treatment. So the next question is, do we see an increase in any of these markers during mastitis? And we looked more specifically at subclinical mastitis. So yeah, we in this case we identified 15 cows that had subclinical mastitis through elevated somatic cell counts and presence of bacterial infection. So we did the vitamin D treatment again, and this time rather than just a one-time treatment, we repeated this treatment for every 12 hours or after every milking, and out to 48 hours. And then isolated somatic cells out of that milk from those treated quarters and measured these responses as a parameter for the immune function. So here's the kind of overall the key results that we looked at. Again, looking at these measures of nitric oxide production and the beta defensins and whether or not they changed in response to the 125B treatment. So here we can see there's three of them that we found, or two of them that we found a main response in, and that's the nitric oxide and another one of the beta defensins, beta defensin 4. And just focus in on that nitric oxide a little bit more. Here, so we gave treatments here at 0, 12, 24, 36, and 48 hours, and then collected the cells every 12 hours after those. And you can see that the 125B treatments here increased the nitric oxide synthase expression during that time compared to the control. So we have a fairly robust response there. And one of the key things that we can get out of this is, earlier what we'd seen, what we'd done in the laboratory, we can actually see this taking place in the cow now. Same thing for this beta defensin 4. This is an antimicrobial peptide, and we're looking at the expression of the gene for this. And again, here we can see we got an increase in that after we've been treated this cow for repeated treatments here. So the kind of the key take home from it, or key message for you guys to get from this, is that yes, vitamin D directly is involved in manipulating that immune response. We're able to use that to enhance the immune response. And really, what are the implications for this? One, we know that for us, we know that vitamin D is important for that immune response. Then, and this has implications for determining dietary requirements for cattle. This has led to actually another project that we have, current milk checkout project that we have going on. And can we use dietary manipulations to somehow increase these markers within the cow? The other is, are there any therapeutic potential for this in terms of using these types of this type of approach to directly enhance the immune system of the udder during mastitis? So it's not something we can take out and use right now, but it's definitely something that has some good potential for the future that we're going to keep working towards, and hopefully be able to develop some alternatives for mastitis therapy out there. So the other project I'm going to tell you about is related more towards the hypocalcemia that takes place in the transition cow, that takes place just postpartum. So as I mentioned, vitamin D, and you guys may have heard this before, vitamin D is well known for its role in maintaining calcium. So this active form of vitamin D, it stimulates uptake of calcium in the intestines. It stimulates retention of calcium within the kidneys and resorption from the bone. And what we found then is that calcium is very critical from activation of the immune response, for that cell to be able to detect and respond to the pathogen. So as you guys are probably aware, during the postpartum dairy cow, that transition that takes place there, there's going to be a drop in calcium. So normally, the amount of calcium that's circulating throughout the blood of the cow is very tightly regulated, right around 9 to 10 milligrams per deciliter, and it's very tightly regulated in all mammals. But one of the problems that the dairy cow faces is during at the onset of lactation, all that calcium that's needed for milk production. So you have a big shift of calcium that's within circulation there to the mammary gland, and the ability of that cow to maintain that is impaired. We can employ the best management strategies. We can put in a negative DCAD diet. We still reach this point where we have subclinical hypocalcemia that's taking place. So the implications of that low calcium, one are if you have severe hypocalcemia, you're going to have a down cow. But really the kind of the overlooked aspect of this is during subclinical hypocalcemia, where you don't have severely low calcium, but it's just low enough, is that the immune system is impaired during that time. And Dr. Santos has done a great deal of work on this in the last few years, showing that subclinical hypocalcemia impairs the immune system and leads to increased incidence of metritis. So what we wanted to do through this is can we use, since we know vitamin D is involved in maintaining the calcium within the cow, can we use that to increase maintain the levels of calcium postpartum? So this is just data showing, I touched on that immunity is impaired in the transition cow. So this is days postpartum. So here's cows with normal calcium, and here's a measure of their immune function, oxidative capacity of the neutrophils, one of the immune cells in the cow. So normally you get a drop in there, but if cows have subclinical hypocalcemia, so a drop in calcium, but it's not severe enough to cause milk fever, you can see that there's a drop in the immune function of those cows. And then here's the data from Dr. Santos' work showing that as you drop calcium further and further, so this is the change in serum calcium in the first 3 days in milk, and as that, so coming this way, as that calcium drops further and further, you increase the probability for metritis. You increase the risk of metritis. So we want to be able to minimize that those negative aspects of hypocalcemia as much as possible. What are the solutions to hypocalcemia? And one, it's not simple. Control of sustained circulating calcium is very complex. It's not just as simple as giving that cow more calcium during that time. Like I mentioned, that the control of calcium is very tightly regulated. It requires increases in the absorption of calcium in the intestine, increases in calcium mobilization from the bones, and also calcium retention in the kidneys. Well, it turns out this is where vitamin D comes in. The active form of vitamin D increases the capabilities of each one of these, so it's able to really effectively increase circulating calcium concentrations. So under normal conditions, whenever you have low calcium, it stimulates the production of this 125D, the active form here. However, in the postpartum dairy cow, this is somewhat impaired. So you're going to have, even with good management, you don't get efficient production of this active form of vitamin D. So the question we had then is, can we come in and treat cows directly with this active form of vitamin D and increase their calcium levels, and subsequently increase immunity? So this is a project, a milk checkoff project, funded primarily to Dr. Santos, and Achilles, his graduate student, was really the one that accomplished this work here. So it's looking at the use of vitamin D, or calcitriol is another name for it, to prevent subclinical hypocalcemia, or vice versa, maintain calcium and improve immune function in dairy cows. So in this project, we took 50 cows. They were fed a negative DCAD diet, so they didn't have, these cows weren't developing, the goal is to not have cows that have severe hypocalcemia, but they would develop a subclinical form of hypocalcemia. And then they're randomized to receive subcutaneous treatments of either a placebo or a 300 microgram treatment, of a one-time treatment of the active form of vitamin D. So these treatments were then given within 12 hours after calving, and then we measured responses to feed intake, milk yield, energy balance, blood minerals, and neutrophil function. So I'm going to focus mainly here on the response to the blood minerals, calcium being the main one, and then neutrophil function of the dairy cow in response to this treatment. So here's the blood minerals and the response that we have of them to the 125D treatment. So here the open circles here are the placebo, are the control treated cows, and then the filled circles are the 125D treated cows. So we have measures of ionized calcium and total calcium here. The ionized calcium being like the free form of calcium that's circulating in the cow. So you can see we have an increase in that, both the ionized calcium and the total calcium. And then the flip of that is magnesium, which we know normally goes down as calcium will increase. We actually get lower magnesium concentrations in these cows. But then we also have an increase in phosphorus. So 125D also actually increased circulating phosphorus. So we also see an increase in phosphorus there as well. So focusing in on the total calcium here a little bit more, we see within during this point, right after calving, we still have cows at this level of calcium would still be considered subclinical hypocalcemic cows. But with the 125D treatment, we very quickly increase the calcium concentrations within those cows. And that's maintained then over several days within this real critical time period here within the first few days postpartum. We do end up seeing a drop in calcium here out to the later days, and really that, in my opinion, this lower calcium at this point compared to the control treated cows here is may not really have a significant impact on the physiology of the cow, but we still have some work to do to really minimize the difference here out at this time point. So we still have work to do to maximize or optimize the dose, the timing of these treatments. But the key point here is we have this increase in calcium within these first few days postpartum, which is very critical in that time point. So next question really that we wanted to get to then, do we see an increase in immune function? So here's some measures of neutrophil capabilities. One is phagocytosis, the ability of that neutrophil to take up and engulf bacteria. And another is the oxidative burst, and that's the ability of the neutrophil to produce these oxidative compounds that are lethal to the bacteria. So within the first few days postpartum then, here again looking at the closed circles of the 125D treated cows, open are the control treated cows. The percent of neutrophils that are undergoing phagocytosis, there was no treatment effect there. But here the percent of neutrophils undergoing oxidative burst, we see a higher percentage of them in the treated cows. So that's a very important critical time period there to have an increase in there, and that corresponds to the increased calcium that we have. The other is looking at, so we didn't see a change in the percent of phagocytosis, but the phagocytic intensity, or the amount of bacteria that they're able to take up. And so here again we see an increase within the first few days postpartum in response to that 125D treatment. And then the other is not just the percent of oxidative burst, but the intensity of that oxidative burst. And same with phagocytic intensity, we have an increase in response to that 125D treatment. So really the kind of the overall conclusions from this is that in this experiment we had this 300 micrograms of 125D given subcutaneously within very close to parturition, so within 12 hours after parturition. That prevented subclinical hypocalcemia, and more importantly, we see this increased neutrophil phagocytosis and oxidative burst. So we see an enhanced immunity through the use of these treatments. So what are the implications from this? One key point is improved calcium as a result of this treatment. That can enhance immune function, and really this we expect this to lead to decreased disease incidence in transition cows. And really what we hope to have from this, not right now, but hopefully fairly soon in the future, a potential adjunctive therapy to using good management, good nutrition, to be able to help minimize the incidence of subclinical hypocalcemia and ultimately transition cow diseases. So real quickly, so this is information that you can take home and use. And one of the questions I often get from nutritionists or dairy producers, are my dairy cattle getting enough vitamin D? And really to answer that question quickly, yes, they are. Most nutritionists recommend around 30 to 50,000 units of vitamin D given to cows, lactating cattle on a daily basis. And we've done some sampling of dairy cows, and we measured one of the metabolites of vitamin D, the 25-hydroxyvitamin D. That's what we use as a measure of vitamin D status to know whether or not the animal has enough. And most of the cows lie within this 60 to 80 nanogram per mil range. And that's actually right about where if you had a beef cow out on pasture, they'd be right in that same range. And we've actually found, we have some data showing that we can increase the amount of vitamin D that they're getting, and you're not going to see any increase in serum 25D over here. So really there's kind of a max benefit. So if you're anywhere in between 30 to 50,000 units, you're doing just fine for your lactating dairy cattle. There's nothing to change there. Here's the other quick point to make though, and especially knowing that vitamin D is critical for immunity, and that's in the dairy calf. And we've done some work in this, and thanks to several of the producers that allowed me to come out and sample some of their calves. And that's dairy calves that get some either milk replacer or milk enhancer. That's going to provide them around 5,000 to 6,000 units of vitamin D per day. And that gets them within the target range for serum 25D for the dairy calf. However, and this is an important point because there are several producers out there that are just supplementing waste milk or whole milk with no added vitamin D supplement. And this is data from our calves at the dairy unit where they're not allowed any access to sun. They're under a shaded structure, and they're not given any supplemental vitamin D. They're fed pasteurized waste milk. And all these calves are deficient through the first few weeks of life here. So that's just an important point to keep in mind, that if you're using that management practice and our knowledge that vitamin D is also critical for immunity, that make sure that if you are feeding pasteurized waste milk, that you provide some sort of vitamin D supplement to those calves. So quickly to wrap up here, just to summarize the key points of the earlier work we had done. So one, intramammary 125D treatment boosts immunity of the udder. And then the other one is that this postpartum subcutaneous 125D, through a different mechanism, through calcium, it boosts the neutrophil function, also boosts immunity. So what do we get from this? We have better knowledge of what factors control the immune system and contribute to a strong immune system. And we hope to be able to use that to provide solutions to transition cow diseases. So with that, a big thanks to the Southeast Milk Checkoff Board for funding these projects. Also want to point out the students that carried out this work, particularly Catherine Merriman in my lab, and then Achilles in Dr. Santos' lab that really carried out this work. A big thanks to them, along with all the other students that contributed to this. So with that, thank you, and happy to take any questions.